Okay, Year 12s, this is it. This is our last bit of content for Unit 4 and for VCE Biology. Um, there will be a bit of recap we'll do on things like practical investigation for a final assessment at the start of Term 4. But in terms of new content, we're onto our very last lesson now. So in the last kind of 2 weeks, we've been looking at biological knowledge, or basically how scientists use biological knowledge for identifying and creating things. We looked at genetically modified organisms in the last lesson. We've looked at gene cloning using bacterial transformation. And we also looked at some of the social and ethical issues around those things. Our final topic, and it's probably never ever been more relevant in the study design, is strategies that deal with the emergence of new diseases in a globally connected world. So looking at that, you're probably thinking this sounds pretty familiar. So the content in today's lesson should hopefully make a bit of sense just based on everything you've been exposed to this year. So it's really important to note, I guess, that diseases and disease outbreaks have occurred since human history started. But we're kind of in a situation now for the first time ever, really due to our understanding and technology, to have more ability to identify and prevent infectious diseases. Okay, so all the things that we've had to put up with this year are done so that we can stop huge amounts of deaths happening, where in the past that would have just happened. A little bit of this will be revision at the start here, but it's worth recapping now. So we're looking here at infectious diseases or transmissible diseases, so diseases that can be spread and passed on, that are caused by pathogens. So remember, a pathogen is a disease causing agent. We used to use the term microorganism, but we prefer agent now because not all pathogens are actually living. So these are things like bacteria, viruses, fungi, protozoa, and parasites. All of these we looked at in a fair bit of depth in Unit 3 when we looked at immunity. Okay, so we're just kind of recapping that because these are what cause infectious diseases. It's important to note that pathogens, usually exist for very long periods of time before causing disease or before becoming an issue. They don't just appear out of nowhere generally. It's just that they start affecting us. And two really common reasons that this might happen is that the pathogen may mutate, so its DNA may change or its RNA may change to become more virulent. Virulent just means, I guess, deadly or dangerous, particularly in the context of pathogens. Okay. Another reason that they might become an issue is that they may jump from an animal host to a human host. And that is quite common. That is what is understood to have been what's happened with coronavirus, that it originally was living in bats for a long time, and we didn't really know what was there until it passed into a human host. Okay, so these are the two common reasons that pathogens can cause sudden outbreaks of disease. Again, this is a quick recap. It's important that you remember or you understand how different pathogens spread or transmit. Okay, so some pathogens are spread through direct physical contact. So that is touching an infected person. That includes sexual contact. Okay, so STIs would be considered a form of direct physical contact. Then there is indirect physical contact, which is touching not a infected person, but a contaminated surface. So something like touching a doorknob that an infected person has touched. Droplet contact, you know all about this one. This is one of the main modes of transmission for coronavirus. So coughing or sneezing. So each of these little droplets has bits of the pathogen in there. This is different to airborne transmission, which is when the pathogen can remain in the air for long periods. And it is thought that coronavirus is possibly in this category. Obviously, it's a new disease, it's hard to know exactly, but there's evidence that suggests that beyond just sneezing or coughing on someone, that it may be able to exist in the air for a certain amount of time. Water contamination is a very, very common cause of disease transmission. Food contamination, particularly meats, which is why it's really important that food is cooked and stored properly. And then finally, vector transmission. We've just looked at plasmids as being a vector for genes. A vector is anything that carries something. So vector transmission would be when, for example, mosquitoes carry the malaria protozoa. Okay, so it's not actually the mosquito that makes a person sick, it's the pathogen that they carry from person to person. All right, a few terms in here. I'd say these are probably fairly familiar, again, based on this year, but these are specified in the study design. Epidemiology is a good word to know. That basically is the branch of science that deals with the study of diseases. So looking at things like the incidence, so the numbers, how many people are affected by something or how it's spreading, where it's spreading to, what is causing it, things like the pathogen, and then how do we manage or prevent or control that. We could say that 2020 is the year of the epidemiologist. They've never been busier with trying to track what's been happening globally this year. Okay, some specific terms that you need to know. Endemic, the term endemic refers to the normal or regular or stable incidence of a disease. In an ideal world, we would have no disease anywhere, but that's not the case. But if a disease is kind of managed at a pretty standard level, we call that being endemic. An example of that is chickenpox. We're not getting any huge outbreaks of that now because of things like vaccination, but there's always a few cases here and there. They're easy to identify. They're generally pretty easy to manage. You see on this map, we've just got kind of some scattered things around. That's kind of just the normal amount of that disease. An outbreak is when we get an unexpected infection of a large number of people. And obviously that's very familiar this year. There are two ways that an outbreak can be classified, and they are an epidemic, which is a sudden increase in the occurrence of disease amongst a specific population in a specific location. Okay, so if we look at this map, if we had a big outbreak in one area, so it could be in one city, it could be in a country, it could even just be like on the border of a couple of countries, but it's very localized. That is an epidemic. And then you should be fairly familiar, unfortunately, with this term this year. A pandemic is an epidemic that has now spread to multiple countries and or continents. Okay, so now we've got these outbreaks all over the world. That would be a pandemic. Okay, so an epidemic can become a pandemic. Doesn't always, but it can. Important definition, this has come up before in the exam, and I'd say there's a good chance it could come up again this year. You don't need to memorize any specific outbreaks or pandemics, but I think it's quite interesting to look at. This is an infographic of some of the deadliest pandemics in history. I think as well this can be good for a little bit of context, that obviously a lot of people are really worried about coronavirus, and we should be, but also in the context of pandemics, it is being extremely well managed. Okay, so in its time, smallpox killed over 300 million people worldwide. Measles has killed 200 million. The Black Death or the Black Plague killed 75 million people, and that's hundreds of years ago, so a huge part of the population. Even the Spanish flu, which was in 1918, so 100 years ago, 50 to 100 million people died from that. Okay, so I know a lot of people have died from coronavirus, but in the context of pandemics, it hasn't been many because of the things that we're able to do now. It is worth knowing some of the factors that can contribute to outbreaks. So remember, like I said, pathogens don't appear out of nowhere. They're generally existing for a very, very long time, and then all of a sudden they either jump into humans or they become more of an issue, and then they spread. So some of the factors are poor hygiene and sanitation. Okay, this is a huge issue around the world. We're extremely lucky in Australia that it's not an issue here. But if you're someone that's doing health and human development, you know all about this. Having poor hygiene, having unsafe water, having a lack of garbage disposal, all of those things increase the likelihood of diseases being transmitted. Densely populated areas. Okay, so having lots of people living in cities or living close to each other makes that human-to-human transmission so much easier. Okay, and that was a big contributor with coronavirus. Wuhan, this is Wuhan here, is a big city. Lots of people living very close to each other, so diseases spread very easily. Adding to that, the increased travel between areas. We live in a very globally connected world, and again, that means that this transmission is so much easier. Again, this was a huge contributor to coronavirus spreading. It happened during the Chinese New Year when people tend to travel all around the world to see their families, so it made the spread of it and it becoming a pandemic much easier. Climate change is a big one. I can already see Jim shaking his head going, oh, here she goes again. But climate change means that we are interfering with habitats where pathogens live, okay? Pathogens live in things like bats, okay? Usually humans don't have that much contact with bats, but if we keep destroying habitats, those environments for those bats get disturbed and they start moving around and they come into contact with humans. This here actually is a bat cave where Batman lives. No, it's a bat cave in Asia where they know that there are probably a lot of viruses and they study these to see what might be coming up that could potentially impact humans. Another issue with climate change, warming temperatures. A lot of pathogens love warm temperatures. They thrive in those temperatures. Okay, so climate change has a big impact on these things. Another thing that can impact how easily a disease spreads is more to do with the behaviour of it and its incubation period, which is the time between getting infected and getting sick or having symptoms. And you probably understand that now. You know, coronavirus, they reckon it's about 7 to 10 days incubation period, which means you can get infected, you don't know you're sick, you're going out and about, you're interacting with people and you infect them. Okay, so a long incubation period means you are more likely to spread the disease because you don't know that you're infected. And then a last one, which we won't go into too much, but it's a really important thing, is the overuse of antibiotics. Antibiotics, hopefully you remember from Unit 3, kill bacteria. They're one of the most important medical discoveries of all time. They've saved millions of lives, but they are being overused. People use them too much now. And the problem here is that some bacteria are resistant to antibiotics or immune to antibiotics, which means that if we overuse antibiotics, we kill the sensitive bacteria. So we kill the ones that we're able to treat, and then the resistant ones survive and reproduce. And that creates what's called superbugs. So I guess it's a great example of natural selection. The sensitive ones die, the resistant ones survive and thrive, but the problem here is we're now ending up with these bacteria that we cannot treat. Okay, so that's a big issue. Okay, this is this is specified in the study design, that you need to know how scientists identify pathogens. So particularly if there is a new disease outbreak, we need to know what is causing it. Okay, it's crucial to understanding the behavior of it and then managing that disease. A couple of methods that scientists use are either physical methods. This would generally be the first thing that is done. You get a sample, you put it under a microscope, probably an electron microscope, because remember we're dealing with tiny, tiny, tiny particles here, especially if they're viruses, and you study what it looks like. Okay, this here is the Ebola virus, looks like a worm, that is Ebola. A lot of viruses or bacteria will look quite similar to other ones. So that would be a physical method. But then the newer technology allows these molecular methods. So looking at the molecules or the genetic material usually. And two common methods of that are amplification-based detection, which is using our PCR, our polymerase chain reaction, to amplify, make lots and lots and lots of copies of a bit of genetic material, so we can sequence or look at that DNA from that pathogen. Another molecular method would be whole genome sequencing. So this would be getting the genome or the DNA of an entire pathogen, and then scientists will look at every component of that. So if we've got here the genome, this is the entire length of DNA of that pathogen. From there, they can look at these individual genes, so they can see what genes make up that pathogen, and from there they can identify the proteins that those genes make. This is, I'm just going to link up to this top picture here. This is the coronavirus virus. That's what it looks like. All of these things on the outside, these receptors, they are proteins. Okay, everything that makes up that shell are proteins. So if scientists can understand the genes that make up these proteins, that can help in studying its behavior and also help with things like trying to create vaccines against it. Okay, again, this is a big focus in the study design, that you can identify or come up with strategies that deal with disease or prevent the spread of diseases. It's really important to note that there is no one-size-fits-all approach with this. Strategies need to be appropriate to the specific pathogen, the way it is spread, and the situation. Okay, a lot of this will be very familiar again based on this year. The first goal with new disease is always going to be to prevent it spreading. Okay, again, that might be different based on what the disease is, but it can be prevented through good hygiene, washing hands, decontaminating surfaces, educating people about the disease and how it's spread, things like proper food preparation if it's a food-based pathogen, clean water, again, if it's a pathogen that spreads through that, and vaccinations. Really, 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 really important thing to note about vaccinations, and this is why I've put it in bold. Vaccinations only exist for diseases they have been made for, okay? So known diseases. So as you know, there is currently no vaccine for COVID, which is why there is such a big focus on the other things of preventing its spread, because we don't yet have a vaccine. Make sure you understand that, because I don't want you writing in your SAC or in your exam, yep, just give a vaccination to the whole population. If it's a new disease, that has not been invented yet. Another important thing that is done, again, this is very familiar, are things like quarantine or isolation. So this ensures that infected or people that are suspected to be infected don't infect other people. This can also be done, it's not just with people, this can be done with animals. And Australia has very strict quarantine laws because we're an island and we can prevent things coming in. Animals, it can be done with food and crops. It's often even just done with shipping containers. Okay, so often things will have to wait for 2 weeks before they are unloaded in case there are diseases being carried on there that could cause infections. Then we'll look at controlling the method of the spread. Okay, so if we're looking at a malaria outbreak, you'd be looking at controlling mosquitoes. So things like bed nets, mosquito repellents, getting rid of stagnant water. If we're dealing with a droplet transmission like coronavirus, things like face masks, or making sure people stay a reasonable distance from each other. Limiting travel, especially for highly contagious diseases. If we're looking at, say, a HIV outbreak, using things like condoms to make sure that that spread doesn't occur. Same for bloodborne diseases, so things like HIV or hepatitis, ensuring that people aren't, for example, sharing needles, which can spread that, or making sure that hospitals have sterile equipment. Okay, so controlling the method of spread needs to be specific to that actual disease. Then scientists will get to work on identifying the pathogen. Okay, so while some experts were sorting out quarantine and isolation, some were figuring out controlling the method of the spread and creating all these rules that we need to follow, other people have been working on identifying the pathogen and finding out as much as possible about it through things like the genome sequencing or the PCR. And then the final step is treating the disease. Okay, prevention is always the first goal. We want to stop it spreading and then ensure that people who are already sick can be treated. The study design this year has removed that you need to know how these treatments work, but I think it's still really important that you understand just these two key things. Antibiotics are only for bacterial infections. Okay, they generally need to be a specific antibiotic for a specific infection. And remember, I mentioned before the risk of overusing antibiotics. There's a lot of issues associated with that, which is why generally they are prescribed by a doctor. Antiviral medications, as the name suggests, are only for viral infections. Antibiotics will have no impact on a viral infection. Antivirals will have no impact on a bacterial infection. So in the textbook, you'll notice there's a whole section on both of these and how they work. You don't need to know that, but you do need to know that antibiotics can treat bacterial infections and antivirals can treat viral infections. All right, for today's worksheet, there's just a few questions that summarize some of that key information. But what I want you to do at the end of that is I've given a few different outbreaks here. I want you to do a bit of research. Just use Dr. Google and complete the table. Okay, so I've done the first one for you. Just note, I want you to look at what strategies you think would be best. So you won't necessarily find that information, but based on the information that you find out about each of these diseases, what would you do if you were the head epidemiologist and you were giving recommendations to the health department on how to prevent this disease? Okay, so I've done the first one for you. The outbreak is AIDS, so the acquired immunodeficiency syndrome. The cause of AIDS is the human immunodeficiency virus, or HIV. The mode of transmission for this pathogen, there's multiple modes. One is sexual contact, one is blood contact, and another one is mother to baby transmission, either during pregnancy or breastfeeding. When I say status, I mean, is it a pandemic or an epidemic? And then the impact. So just don't spend too much time on that, just a little bit of data. So AIDS is certainly classified as a pandemic. It's spread all around the world and has infected a huge amount of people. Over 65 million people have been infected since it was first identified in the early 80s, and over 30 million people have died. Strategies based on this mode of transmission would be educating people about the disease and how it's spread, ensuring provision of condoms to people, having sterile procedures in hospitals, anywhere where blood is involved, using single-use syringes. So, for example, for IV drug users, ensuring they have safe injecting practices. Early diagnosis and treatment. HIV can't be cured, but it can be treated. You can stop it spreading. And then things like if a mother is infected and knows she's infected, having alternatives to breast milk. So ensuring she has formula available so she doesn't transmit that to her baby. Okay, so I want you to do a bit of research on SARS, Zika, tuberculosis, and Ebola, and just fill out that table. All right, thank you.